Indo-Pacific sailfish. So let's go over taxonomy and classification. So kingdom Animalia, phylum Chordata, class Actinopterygii, order Periciformes, family Isidoridae, genus Istophorus, and species Istophorus platyparus. All right, so who named this species? Well, it was named by Shaw and Nodder in 1792, the common name Indo-Pacific sailfish, and the family Istophoridae includes marlin, sailfish, and spearfish. Little species debate. The genome-wide analysis confirms two distinct populations, one Atlantic and one Indo-Pacific, that diverged 1.81 million years ago during the early Pleistocene, and they are genomically distinct but not formally recognized as subspecies. But some do, some don't. A little scientific debate between scientists. Physical description. So the sailfish are dark blue dorsally, brown blue laterally, silvery white ventrally, and they have stripes of light blue dots which run along the lateral sides and have a massive dorsal fin. Max length is 430 or 340 centimeters, max weight 100 kilograms, and the lifespan is around 13 to 15 years with a burst speed of 110 kilometers per hour. So sexual dimorphism, females are slightly larger than males on average, which is a common trait in billfish, but it's also worth noting that females grow faster throughout the juvenile stage. Anatomical structure. So these fish are fusiform, laterally compressed, and highly streamlined. The scales become nearly absent in adults because over time they just grow smaller, and this also reduces surface friction. So the lunate tail provides powerful thrusts with minimal drag. The bill is not used to spear, but rather to slash and stun prey by rapidly turning the head. And the jaw is lined with file-like teeth that grips stunned prey. Chromatophores. So these are pigment cells which allow rapid color changes, and they flash patterns for communication during breeding and to coordinate hunting behaviors. And those are the light blue dots that I mentioned earlier. So these fish have a lateral line which runs curved above the pectoral fin and straight to the tail, which detects movements like pressure changes. Which detects movements and pressure changes. They have large eyes that flush at the head, and they are sensitive to low light. They also have nares, which detect dissolved chemicals. The sail or dorsal fin is densely vascularized and is also theorized to regulate blood temperature before and after high-speed sprints by raising or lowering the fin to expose blood vessels. Life cycle. So the eggs first start at 0.85 millimeters, and then they contain oil globule for nutrition, and they hatch in around 60 to 70 hours. And once they are fertilized, they average around 1.3 millimeters. They are 1.96 millimeters at hatch when they are larvas, and they reach 2.8 millimeters by day 3, 15.2 millimeters by day 18, and the bills begin to form pretty early on. At the juvenile stage, they will experience exponential growth in year 1. The sail and bill will become more prominent. The schools will be in size cohorts, and as adults, they become adults after 3 to 4 years, and they mature at around 162 centimeters. By then, they will have full coloration and sail development. The females will be noticeably larger, and their growth will continue lifelong. So their lifespan is around 13 to 15 years, but the catch and release specimen average around 4 to 5 years. And also no parental investment at any stage. Habitat and ecosystem. So their range is in tropical and temperate waters of the Indian and Pacific Ocean. Their depth zone is the epipelagic zone, around 0 to 200 meters, and they stay near the surface where sunlight penetrates and prey concentrates. And they will occasionally dip to 350 meters. The ecosystem they live in is the open ocean epipelagic ecosystem, which is filled with schooling forage fish like sardines, anchovies, and mackerels. It is also home for tunas, sharks, dolphins, and seabirds above the surface. So some hotspots, you'll find them concentrated near upwelling zones and thermocline fronts. And the temperature they like is warm water currents above 28 Celsius. Diet hunting and natural threats. So their diet includes mackerels, sardines, anchovies, cephalopods like squids and octopus, occasionally neretic and benthic prey, and the same size juvenile targets the same prey. So body size, not age, determines diet. Hunting. Solo or in groups, they avoid their folds sail flat, or they fold their sail flat to reach the 110 kilometers attack speed, and they rapidly turn their bills to stun and slash prey. And their chromatophores flash coordinating group attacks. Some of their predators include pelagic sharks and orcas. Some defense adaptations. So they have counter shading, which provides camouflage from both above and below. And due to their fast speeds, they can escape predators quite effectively. Migration. So they migrate seasonally, and they migrate annually to higher latitudes in summer, following oceanic currents above 28 Celsius, and then they return towards the equator in autumn. And during spawning season in the Western Pacific, the mature fish migrate from the East China Sea southward toward Australia. A genome-wide analysis, as mentioned earlier, confirmed two oceanically isolated populations, the Atlantic and Indo-Western Pacific, but there's no transoceanic migration that occurs between them. The migration is dependent on sea surface temperature, and the sailfish constantly track warm water fronts above 28 Celsius and avoid cold upwellings. Interspecies relationships and symbiosis. So some known parasitic symbiotes include Panella instructa, Calitarianiscus gracilis, and Forceps menicithus. And all these parasites affect the stomach wall. The parasite effects haven't been studied too thoroughly yet, though. Some competition includes other large pelagic predators like tuna, marlins, and mahi-mahi. Commensalism. So drone footage off Malaysia documented terns, which are seabirds, or a species of seabirds, attacking bait balls immediately after sailfish strike. By scattering and disabling prey, sailfish create surface foraging opportunities for the seabirds to exploit. The sailfish gain nothing, so the relationship is commensal. Within species relationships. So juvenile schooling. These are the cohorts, as mentioned earlier. So juveniles swim in dense schools structured by size because cohort grouping reduces predation risks. Also, trophic overlap studies show that juveniles of the same size class target the same prey, suggesting that cooperative size cohort foraging. Suggesting cooperative size cohort foraging. Adult social structure. So these fish, they have no home range or territory, and individuals are highly mobile and do not defend space, and groups form opportunistically around prey aggregation. Communication. So their primary form of communication is visual and is done through chromatophore-driven color flashing during breeding, group hunting, and dorsal fin movements can be signals of aggression or submission. Reproductive strategies. So they do polygynandrous broadcast spawning, and multiple males court a single female, and fertilization is external with no parental investment whatsoever. They breed 3 times a year, spring, summer, fall, in the thermocline layer, and they spawn year-round in tropical regions, spawning migration to Western Pacific and East China Sea. Sorry, spawning migration to Western Pacific and East China Sea occurs in mature fish. Around 800K to 1.6 million eggs spawn per spawn, and 1.2 million average offspring. 60 to 70 hours is the hatching time, and 3 to 4 years is for sexual maturity. Evolutionary history. So the around 5.33 million years ago, in the Pliocene era, the modern billfish lineages diverge. So Istophoridae splits into marlin, sailfish, and spearfish lineages during the major ocean reorganization. 1.81 million years ago, in the early Pleistocene, two sailfish lineages split. That is the Atlantic and Indo-Western Pacific populations, which diverged during glacial isolation. This is the origin of today's two genetic populations. And then 80 to 10,000 years ago, the both populations showed bottleneck signatures from repeated glacial refugia events and low average genome-wide heterozygosity, which reflects these historical reductions. And then in the 1950s, industrial fishing begins and a population crash happens. So abrupt effective population size drops 80 years ago, and in the Atlantic, an 80-fold reduction, and in the Western Pacific, a 10-fold reduction. Human impacts. So commercial bycatch. So they are primarily taken as bycatch by tuna longliners in the Indian Ocean, but they're also caught by gill nets, trawling, and harpooning. An estimated 28,800 metric tons is caught in the Indian Ocean in 2000 alone from just commercial bycatch. So according to the Indian Ocean Tuna Commission, this species is classified as data poor, and the average annual catch from 2008 to 2012 is 26,283 metric tons. Direct harvest. So meat is valued for sashimi and sushi in Japan and Southeast Asia, and sailfish are a major sports fishing target, which drive economic activity, but also catch and release mortality. And as you can see, that photo is of me when I caught a sailfish in Mexico, which tasted great, gotta say. So population status and threats. So according to the IUCN, they are vulnerable, and all populations of sailfish show trends of decline. And this is because of overfishing, because the species is sought out for its meat and for sport, also caused by bycast, like longline tuna fishing, and mismanagement. So the Atlantic and Indo-Pacific populations are genetically isolated and can't replenish each other. But the RMFO classifies them as one, which makes recovery much harder if either is overfished. Also, the population is vulnerable because of climate change, like ocean warming, deoxygenation, and shifting prey distributions add cumulative pressures. Habitat loss, coastal development, and pollution degrade juvenile nursery areas. So these are two of my research papers. The recent one, the paper written within 5 years, Seascape Genomics and Phylogeography of the Sailfish. So this research paper is an in-depth genetic analysis done on these sailfish. The goal of the study was to figure out why there are so many distinct populations of sailfish that exist, and they went on to look at different environmental factors such as salinity, temperature, oxygen levels, etc. Most interesting of the research papers I saw, well, that is the sailfish generate foraging opportunities for seabirds in multi-species predation aggregation. So this research paper was very interesting because it used a drone to study the relationship between terns and sailfish off the coast of Malaysia, and it was found that they have a very strange commensal relationship that I mentioned previously. And that is all. Here are my references. Thank you.